To get into a little more details about how this works, we're going to be taking a look at this figure here, and it looks like it's much more involved, but it's still telling the same story. This picture is a nice illustration of the central dogma of biology. DNA is transcribed into mRNA, mRNA is translated into a polypeptide sequence. This is actually showing exactly the same thing. It's just taking like a little bit of a closer look at what's happening here. All right, so let's start off with our triplets. Our triplets on the level of the DNA are going to turn into codons on the mRNA transcript. Triplets turn into codons on the mRNA transcript. So we start off with DNA, we transcribe into mRNA transcripts, and the triplets will then cause sort of like the mirror image of the original triplet on the mRNA. So TAC becomes AUG, GGT becomes CCA, because when we create an mRNA transcript, it's going to be complementary, or like the pairing, the typical pairing that you would see from DNA to RNA. So here we have our DNA. We have our original DNA, which was a double helix kind of tied together. And here we're showing that the RNA polymerase kind of unzips it and starts to create this mRNA transcript. And we take a closer look at this, what we'll see is that every time you have a GACT here, it's going to create its opposite pairing on the mRNA transcript. And that is how, whenever you have a T on the original DNA, it's going to create an A on the mRNA transcript. When you have a C, it'll create a G. When you have a G, you'll create a C. When you have an A, it will create a U. Remember, it's uracil with mRNA. So with RNA, it's a U rather than a T. Okay, so now we have these codons, which are complementary to the original strip of DNA along that gene. That codon then will eventually create a particular amino acid. Here are the amino acids that are eventually going to be peptide bonded together. They're going to be bonded together in that peptide bond, and that will be the job of a ribosome to do that work. AUG, the codon AUG, is going to indicate methionine, or MET. The CCA will create this amino acid, proline. Now, when we look here, the reason we know this to be true is we can look at the genetic code. And so here we have our AUG, which is methionine. We have our CCA here, which is proline. CCA would be CCA. CCA right here, proline. And so we can look at our genetic code and see which triplets on the mRNA transcript will translate into which particular amino acid, and it's faithfully carried out in this way. Now you'll notice we skipped right over this guy here, which is a tRNA. The tRNA is going to have an anticodon, and it looks kind of like this. Here's our anticodon, UAU, will indicate isoleucine, because the tRNA has an anticodon on one side, and it actually has that amino acid on the other end, and therefore it carries out the orders from the mRNA transcript. So this anticodon will be complementary to the mRNA transcript. So UAU indicates isoleucine, and that is because the original mRNA transcript had an AUA. There it is, isoleucine.